Morning and welcome to this course on front-end vision and deep learning. My name is Bart Romani. I'm Emeritus Professor here at Eindhoven University of Technology in the Netherlands. And I have a long career behind me on medical image analysis. I go talk in this course about can we understand what deep learning is actually doing under the hood? And can we relate this to modern findings in biology? What is our visual system doing? We know that the cascade of steps in vision is very similar to the cascade of deep learning neural networks, in particular convolutional neural networks. And in this course, you see to the upper right, we very much focus on the relation between biological vision and computer vision. We have two guest lecturers in this course, Mitko Veta and Erik Bekkers, and they will talk about applications. Mitko will talk about pathology, and Erik Bekkers will talk about orientation invariant convolution filters. So it's a course on deeper understanding of deep learning and human vision, and it is not a course in how to apply deep learning. There are so many courses on how to apply deep learning. If you take courses from Stanford University, from TensorFlow, Café, there are lots of downloadable courses on the internet. If you look for good books, they often come with nice courses. You can download the code on GitHub. It is very nice, actually. You download the code, you put in your data, and it actually works. So this course is not repeating those courses, these fantastic courses that already exist. But we go a little bit deeper and try to understand what do these kernels do? What is contextual processing? What happens if we take color differential structure, or if we have time operators? How is learning actually working? Can we mathematically represent very complex statistical data in a very nice basis so we can have a compact representation? So that's what we try to understand, the black box idea. I'm not claiming that this course will teach you fully understand how it works, because there are still many, many open questions. But it teaches you a style of approach that you say, well, let's not take a heuristic approach, but a detailed approach that we step by step understand more. And today we are in the first levels. We pretty understand what they do, but the deeper levels, it's still very much open, and you have a career to come, and maybe you make some breakthroughs in this area. We go take some mathematical approaches. We use axiomatics, that means reasoning from first principles. And we deeply dive into some recent papers, Nature papers, what has been measured in the brain. There are beautiful optical techniques, nice MRI techniques. There are optogenetics techniques, adaptive optics. We all go discuss these techniques, and you will see that the revolution that we see in computer vision, there's the same revolution in brain research, and we're going to match these two. One can learn from the other, one can teach models from the other, and it really goes backwards. We look at self-organization, and in principle, the approach that we take is very much based on a school that was started a long time ago with Jan Koendering, and which I worked for many years. It's called differential geometry, with people like Luc Verrak, Erik Bekkers, Remco Duits. We did a lot on finding the mathematical geometrical structure of images and the stages in the brain. And everything we discuss, I give you the code, and I use Mathematica. It's a very sophisticated, high-level playground for mathematics. It is really a design language, and I hope you will see the beauty of working with this program in very concise and very related to how we people perceive programs. So it's more related to English than to programming in loops and fine detail. It is really a program on the high level. So we have 4 weeks, every Wednesday and every Thursday. In the morning we have 3 hours of classes, and afternoon we exercise with everything that we have seen in the morning. And the first week will be basics, introduction to deep learning. We give an overview of what is known from the visual system, the latest state of the art, what can be measured. We look at brain connectivity. We look at what is the different layers, what happens in V1, what happens in the thalamus. And we give an introduction to this programming language so you can be more fluent in experimenting. And the second week, we start doing some mathematics of self-organization. Why do these trained networks organize themselves? They, in some sense, they optimize something. They optimize the use of energy, they optimize the representation of data. And we're going to see that actually the visual system, it takes derivatives to quite high order. So we see not only edge detection, but we take second-order derivatives, third-order derivatives, and we're going to see what is the nature of these derivatives. There are multi-scale derivatives at different orientations. So we deeply understand, try to understand the nature and use of these first-stage filters. In week 3, we go deeper with these filters and we say, okay, we not measure intensity, but we measure much more. We measure color, RGB. We measure time. We measure disparity, depth. We have filters that are multi-orientation and multi-scale. So we look at the little bit more sophisticated view on our kernels. And in week 4, we use the knowledge that we gathered so far in applications. So we're going to study a range of very nice medical applications where we use the kernels that we have seen so far. And because we have so many discoveries in vision and in deep learning, but these two worlds, they are quite disjunct. If you ask a computer vision person about biological cell properties or a biologist about a recent finding in mathematics or statistics, they hardly know the different jargon. And it's nice to bridge those two together. So in the last week, I will show you some findings, recent findings in both fields and how they might be related. And I'll give you a nice overview of a big project, retina check, that we did to screen in a large population for the presence of diabetes by studying early damage in the retina, where we used all the properties that we discussed in this week. So that's a nice recap with applications. And in the final hour, we have some questions and we study the whole week. We have some practicalities. There's a website you have all seen. Lectures and computer lab. The computer lab will be in the Matrix building. Dr. Van Huang, who was my PhD student, he will be next to me available during the afternoon. And all information you can find on the website. It's all public. We have PowerPoints, Mathematica notebooks, and a lot of literature. And today we take a group picture just around the corner here to have everyone present here in the picture. We have an exam. At the end, there is a list I'll make available. And you take 3 topics from this list, and you make a nice report on the question that we have there. And you can pick any. They are simple and they're difficult. And I ask you to stand on your toes. Take 3 that you say, okay, that's a challenge for me. And you hand in the results. There's a deadline 3 weeks after the course. It's not a really hard deadline, but you have so many other things to do. Don't spend too much time, and you need to continue again. So try to keep this deadline. And with this said, I start the course and finish this introduction. Thank you very much.